Okay, hello and welcome to the sixth lecture or fifth video about the osmovolume and pH regulation of cells given by Professor Pane. So, first of all, in the beginning of the lecture, you've got some very confusing slides, especially this one with all the numbers and stuff here, and the last one, which is this one. Well, you don't need to memorize any of those numbers. These first lecture slides are here to introduce the Donan effect, and then based on Donan effect, these lecture slides, they want to conclude that cells have to have a pump-leak model of osmovolume regulation. Now, what does that mean? First of all, what is Donan effect? Donan effect is the tendency of inorganic ions to reach thermodynamic equilibrium between different sides of a membrane. So you can see, if this is our cell here, we have an extracellular matrix and a cytoplasm. And the inorganic ions on each side, the concentration of inorganic ions on each side is different. But there is a tendency that these inorganic ions want to reach an equilibrium of concentration between each side. And that results in a net influx of ions from outside the cell, from extra cellular matrix to the cell. Influx. So these ions will leak to the cytoplasm of the cell. Now, as you probably know, water molecules passively follow to a place with higher concentration of ions. That's called osmosis. Osmosis is the diffusion of water through a semi-permeable membrane. And water follows ions. So if ions just continue leaking to the cytoplasm of the cell, a lot of water will diffuse via osmosis to the cytoplasm. Then the cell will swell and will get bigger and bigger to extent that its membrane will rupture and the cell will die. So cells, they need some sort of a mechanism to compensate this leaking of ions. And that's why they have different types of pumps on their membrane, especially sodium-potassium pump, which, if you remember, we talked about it in details in previous lectures, and especially that this pump maintains a very important sodium concentration outside the cell. Now, another thing that this pump does is that it transports 3 sodium ions outside the cell and 2 potassium ions inside the cell. And by doing so, if you sum these charges up, there is a 1 ion per cycle going out. And this 1 ion per cycle going out is compensating these ions coming in. So the net influx or efflux of ions is 0 in case of these cells. And that is a pump leak model of osmoregulation. So you see, ions leak in, pumps in as a net influx take these ions out. That is a pump leak model, but that works in an isotonic condition. So you probably remember the definition of an isotonic condition from biophysics. But again, isotonic conditions are the isotonic condition is a condition in which the concentration of solutes, which means all the ions, all the other stuff here, which are solutes, the concentration of solutes between different sides of the membrane is roughly equal. So what happens if we put this cell in a hypertonic or in a hypotonic medium, which are anisotonic mediums? They are not isotonic mediums. So is cell gonna just swell and rupture, or is it gonna shrink and just deform and then die again? Or is cell gonna find out another ways to regulate, to compensate these changes? well, the second one is accurate, and this lecture, most of it is about that how these cells are going to react when they're in hypertonic or hypotonic situations. First of all, let's talk about cells in a hypotonic situation. Now, what is a hypotonic situation? First of all, we know a hypotonic situation is not isotonic. It is a subtype of an isotonic situation. Now, in hypotonic mediums, the concentration of solutes, the concentration of ions especially, the most important ones are ions. The concentration of inorganic ions inside the cell is way higher than the concentration of ions outside the cell. So what's going to happen is that water molecules will have a tendency to follow the inorganic ions to come in the cell, to diffuse and come in the cell, and then as a result, cell will swell. And by the way, there is another concept that you should know by now, but again, from biophysics, but again, I'm gonna define it again. Osmolarity. Osmolarity is a concept which shows which shows the tendency of water molecules for entering a place. For example, if the osmolarity of the intracellular matrix, if the osmolarity of cytoplasm is higher, it means water molecules want to perform osmosis and go in the cytoplasm. If the osmolarity of extracellular matrix is higher, then water molecules will leave this place and go to a place with a higher osmolarity. And that's about osmolarity, which you need to know. So in case of a hypotonic medium like here, a hypotonic situation like here, the osmolarity of cytoplasm is higher. So water molecules will diffuse into the cell and cell will swell. In our case, these cells are lymphocytes, which are part of the white blood cells. And the cell will continue swelling, and if it continues swelling like this and cell doesn't compensate, the membrane will rupture and the cell will die. But cell's gonna compensate this swelling. And how is that? Well, the cell basically needs to get rid of the excess of water molecules. Now, there are two main ways of doing so. Either by transporting water molecules actively from its cytoplasm to the extracellular matrix. So it means that simply cell would have to have some transporters on its membrane to get these water molecules and just throw them outside. And unfortunately, human cells do not acquire such transporters. Some other cells in other species do, but human cells don't. Now, what they have are ion channels and transporters. And what they're gonna do is that, well, they will transport ions outside of the cell, and water molecules will follow those ions. This is another way of getting rid of water, right? So there is a mechanism which is I think very genius in the cells, that when the cells swell in a hypotonic situation, there is a specific type of chloride channel, a membrane stretch gated chloride channel. Now, if you remember, a membrane stretch gated channel is sensitive to mechanical stress, especially the stretching of membrane. So when the cell swells, its membrane stretches, and these chloride channels will be activated. And when they are activated, they'll open and they will let the efflux of chloride ions from the cytoplasm to the extracellular matrix. And as you can see here, these chloride ions are negatively charged. So we are basically getting rid of negative charge in the cytoplasm. So the cytoplasm of cell is getting more positive or less negative, same thing. And if the cytoplasm is getting more positive, what we're gonna have is a depolarization of the cell. And when depolarization occurs, we again have another specific type of channels, depolarization activated potassium channels or voltage gated potassium channels. So when the cell is depolarized, these voltage gated or depolarization activated potassium channels are gonna open, and then we'll have the efflux of potassium from the cytoplasm to the extracellular matrix. So we are losing ions. Basically, we are losing ions, and when ions are leaving the cell, the osmolarity of the cytoplasm will lower. And when the osmolarity lowers, water molecules are going to leave the cell. They're going to follow the direction of ion flow, and there will be a loss of water, and the volume will decrease. The cell will kind of compensate this hole getting bigger and bigger to extent that it will die. So the cell will manage to survive. Now let's talk about the exact opposite situation. What if we put these cells in a hypertonic situation, which is again another type of anisotonic condition, a hypertonic condition. Well, in a hypertonic condition, first we've got the cell, then as you know, hypertonic is exactly the opposite of hypotonic, so we're going to have a large concentration of inorganic ions outside the cell. So the water molecules will try to leave the cell and cell will shrink. But cell now needs to fight these changes. So cell needs to gain back water, the exact opposite way of hypotonic situation. And how will cell manage to compensate this loss of water? Well, the cell will compensate that by activating a specific type of channel, a specific type of transporters, or to be more specific, antiporters, sodium proton or sodium hydrogen antiporter. The sodium hydrogen antiporter transports sodium ions into the cell, but by the expense of transporting hydrogen ions out of the cell. So you may think to yourself, wait a minute, in order for water molecules to come back into the cell needs to gain ions. Now, okay, by coming of sodium ions into the cell is gaining ions, but at the same time it's losing hydrogen ion. So what is the use? What is really the use of these antiporters? That is because when hydrogen ions leave the cell, when protons leave the cell, the medium of the cell will be more basic, because as you know, hydrogen ions or protons are responsible for the acidity of the cytoplasm. So when they are leaving the cell, the cytoplasm will be less acidic, it will be more basic, and when that happens, another type of channel will be activated. Another type of antiporter will be activated, bicarbonate chloride antiporter. And this antiporter will be activated in order to fight with the pH change in the cytoplasm of the cell. So what we said here is that this loss of hydrogen ion is making the cell more acidic. So the cell is making the cytoplasm more basic, less acidic. So what the cell needs to do is that it needs to get rid of its basicity, so the cell can go back to the right pH. And how does the cell do that? Is by transporting bicarbonate, which is responsible for basicity of the cytoplasm, by transporting bicarbonate outside the cell. So you can see both hydrogen, which is responsible for acidity, and bicarbonate, which is responsible for basicity, are leaving the cell. So the pH will remain roughly the same. But again, this transporter is an antiporter. So while it's transporting bicarbonate outside the cell, it's also transporting chloride ions inside the cell. So what we will have at the end is sodium ions and chloride ions, basically salt, inside the cell. So the concentration of inorganic ions will rise, and when that happens, the osmolarity of the cytoplasm of the cell will rise and water molecules will come back to the cell. And you can see here this whole thing is a cycle, because when water molecules come back here, which is H2O, they will contribute to forming these protons, these hydrogen ions. So water molecules will form a reserve of these hydrogen ions in order for these antiporter to be activated. And well, as you know, CO2 is also absorbed by the cells via diffusion. And when these CO2s are absorbed, they will react with water, and what happens is one of the products will be bicarbonate. So again, cell will have a reserve of bicarbonate molecules in order to transport out, so that these antiporters will remain active. And if you really understand that, you can also understand the pH, regulation of the cells. Because let's assume the cell is in an acidic medium. Let's say we take these cells and put them in an acidic medium. So the cytoplasm of the cell is getting more acidic and more acidic, and the cell wants to compensate these changes. What it does is that this sodium hydrogen antiporter will be activated. The more acidic the medium is, the more efficient, the more rapidly this antiporter will function, and this antiporter, by getting rid of hydrogen ions, will make the will make the cytoplasm more basic again. The opposite is also true. If we put these cells in a basic situation, in a basic medium, what happens is that this bicarbonate chloride antiporter will be activated, and this antiporter will get rid of the bicarbonate ion, and by doing so will compensate this basicity and will make the pH of the cell to remain as we need it. And one more thing that I need to mention about these about cells in hypertonic and hypotonic situations here is that these regulations you saw, well, their names suggest what's happening here. For example, in this case, in case of a hypertonic situation, what cell is doing is a regulatory volume increase, right? And in the other case, in case of a hypertonic situation, what cell was doing was a regulatory volume decrease. So cells are regulating their volume, but both of these regulatory volume increases, which we mentioned, are short-term regulatory volumes. These are short-term. We're going to talk about long-term volume regulations just in a moment. Now, before we talk about long-term volume regulation, we need to know something else. That, as we mentioned, in organic ions, the change in concentration in inorganic ions in the cell or outside the cell can affect the osmolarity of the cell or of the medium in which cell is. So here you can see factors determining the intra and extracellular osmolarity, and one of these factors is inorganic ions. There are other factors determining the osmolarity. The next important factor which we're going to talk about are metabolites. Well, metabolites are these organic material in the cells. So the main difference between metabolites in these cases that we are talking about, in these situations, the main difference is that metabolites, they can polymerize or depolymerize, the thing that inorganic ions cannot do. So what does that mean? Well, let's think we have got 10 metabolites in a cell. Now, these 10 metabolites, they can polymerize, these 10 monomers can polymerize and become one metabolite by binding together and forming one big metabolite. Now, if you remember when we were talking about osmolarity, we said that the concentration of inorganic ions or the concentration of solutes in general determines the osmolarity. And that is also true about metabolites. But bear in mind that the size of solutes has nothing to do with osmolarity. So in our example, when we have 10 metabolites in our cell, the osmolarity of the cytoplasm of the cell is higher. So water molecules are going in the cell via osmosis because there is a higher concentration of metabolites in the cell. Now, during volume regulation, another thing that cells can do along with transporting ions and stuff is that they can polymerize or depolymerize metabolites. So in our example, these 10 metabolites can polymerize and become one big metabolite. Now the concentration of metabolites in the cell will lower and the osmolarity will lower with it. So water molecules will no more come in the cell because the osmolarity of outside the cell is now more than the osmolarity of cytoplasm in the cell. So the water molecules will leave. And that is another genius way of regulating the volume of cells. And as you can see, this lecture slide sums the volume regulation, both short-term and long-term volume regulation of cells. So let's go through it. First of all, we're gonna put cells in either a hypotonic environment or a hypertonic environment. Now, if we put a cell in a hypotonic environment, what's happening, the definition of hypotonic, as we said, is that the concentration of solutes is more in the cell. So water molecules are gonna come in the cell, they're gonna diffuse via osmosis into the cell and cell will swell. In the hypertonic case, the concentration of solutes outside the cell is more, so water molecules will leave the cell and cell will shrink. Now the cell needs to fight these changes. So in case of hypotonic environment, the short-term reaction will be RVD, or regulatory volume decrease, which causes the loss of ions. And how does that work? Well, I'm sure that you remember, first by activation of a membrane stretch-gated chloride channel, which leads to the activation of a potassium channel, a voltage-gated potassium channel, and potassium and chloride ions will leave the cell, so water will leave the cell and will have loss of ions, and as a result, loss of water, so cell will stop swelling. In case of hypertonic situation or hypertonic environment, we will have RVI, or regulatory volume increase, which is accumulation of ions via those two antiporters we talked about, the sodium proton antiporter and bicarbonate chloride antiporter. Now these were, as I said, short-term regulations. What are the long-term regulations? Well, in case of a hypotonic environment, we will have anabolic processes. Now what does anabolic processes mean is, anabolic in general, it means when we have several things and these several things will bind together to form one thing. So basically polymerization. You see here, monomers will become polymers. So the concentration of the metabolites will decrease because we have 10 monomers, then it will become one polymer, 10 metabolites to one metabolite. So the concentration of metabolites decreases, and if that happens, the water molecules will also leave the cell to go to a place with a higher metabolite concentration. The opposite thing will happen in case of a hypertonic environment, depolarization or catabolic processes. Catabolic means when one thing degrades to several other things, or depolarization, when we have one big metabolite, then it will become 10 smaller metabolites, but at the end the concentration of metabolites rises, and so water molecules will follow the higher concentration into the cell. Now cell will survive from shrinking. Cell will get bigger again. Now there's another way to play with these metabolites, except, depolymerizing them or polymerizing them. There's another way to play with them, and that is to transport them. So in case of hypertonic environment, we can get rid of these metabolites. We can transport them outside the cell via these taurine transporters. And in case of a hypertonic environment, we can transport these metabolites inside the cell, again via these taurine transporters. So what these taurine transporters do is that they transport metabolites. Now that we've talked about short and long-term volume regulation, I think it's now time to complete our picture about the pH regulation. So you can see a nice graph here. On the x-axis, it is the pH. So the more we go toward the right, the more basic the pH is. The more toward the left, the more acidic it is. On the y-axis, it is the percentage of maximal transport speed. So the higher you go in the y-axis, the more effective, the faster will the transporter works. And this red line here is indicates the sodium hydrogen transport, and this greenish line here indicates chloride bicarbonate transport. So as we said, if we put cell in a very acidic situation, like here, let's say the pH here is about 2, it's very acidic. When we put cell here, the sodium hydrogen antiportal will be active in its full capacity, in its full speed. Why? In order to get rid of hydrogen ions by the expense of letting sodium ions come in, as we talked about these type of transporters. So it will be functioning in its full speed, and the more the pH of the cell is reviving to its resting state, the more the pH is getting normal, the less the effectiveness of this transporter will be, the less its speed will be. And the same thing about chloride bicarbonate antiporter. If we put cell in a very basic medium, like in a medium with a pH of 7.6, which is very basic, this antiporter will be active, will be functioning in its full capacity, in its in the highest speed that it can be. And what it does is that it transports bicarbonate ions outside the cell. Bicarbonate ions are basic, so when we are transporting them outside the cell, cell is going back to become more acidic, to become more normal. And the same thing as in case of the sodium hydrogen antiporter. The more the cell the more the cell is reviving to its normal state, the lower the effectiveness of this channel will be, of this transporter will be. And you can see an intercept of these lines in this graph. What this intercept indicates is the steady state pH. This black dot indicates steady state pH. What is that? That is when the rate of base efflux from the cytoplasm to the extracellular matrix is equal with the rate of acid efflux from the cytoplasm to the extracellular matrix. And that makes complete sense, Look here. Here the rate of the red one, the rate of sodium, the rate of action of sodium hydrogen antiporter is equal with the rate of action of chloride bicarbonate antiporter. The rate of action of both antiporters is equal, so the rate of efflux of hydrogen is equal with the hydrogen ion is equal with the rate of transport of bicarbonate ion. So the cell will remain in that state, which is a steady state pH. And one more thing about the pH regulation is that there are other transporters working in order to regulate the pH of cells. For example, you can see here chloride sodium or just bicarbonate transporters. Now, I do not talk about them because I haven't seen these ones in any exam. I haven't heard that they've come up in any exam. So the main important ones actually doing the job are the sodium hydrogen antiporter and chloride bicarbonate antiporter. And by saying that, we have finished this lecture, which was about the volume regulation and pH regulation. See you in the next lecture.